Hello, this is Michael Edwards, CEO at Bioinfo Solutions. This video is part one in a series I'll be doing on crowdsourcing coronavirus data. I also want to announce that all the data sets that we'll be using and presenting on in these series of videos will be free and available to the public. So please check out the description of this video and you should find a link to my GitHub account where you can download and access all the associated files. I know I share this sentiment with a lot of my friends and colleagues in science, but when something like the COVID-19 epidemic happens, you feel powerless and you want to help. It's my hope that by gathering hundreds of thousands of dollars worth of coronavirus research data together in one place and some of the biological insights that we've been able to gain from looking at that, that it might help benefit maybe a researcher or clinician on the front line of this epidemic. This next slide is just an outline of some of the topics I'll be discussing in the next couple of videos. To start off with, I'm going to give you some background on SARS-CoV-2, how it infects its target cell, and what are some of the cellular responses to that infection. I will also give you the rationale for why we used a related strain of virus, SARS-CoV, as a surrogate for infection for the current epidemic. I will then explain how we use the Illumina platform base-based correlation engine to collect gene expression data from multiple animal studies looking at the host response to SARS-CoV in the lung. I will also show how we can combine all of this information to generate a single gene expression profile at different time points post-infection that we can probe for biological insight and even therapeutic intervention. And finally, I will explain the metadata files associated with this analysis that are available for public download, which you can use to further your own research or follow along with these videos. Before I get into the meat of my talk, a few disclaimers and disclosures. Basically, all this is saying is that everything you're hearing that's coming out of my mouth is all me and doesn't represent anybody else, and that no one's paid me to do these videos, although I have received plenty of technical help from the individuals that are listed on my title slide. This first figure shows the life cycle of the SARS-CoV-2 infecting its host cell. It is believed that the virus binds to cells containing the angiotensin converting enzyme 2, or ACE2 receptor. A list of tissues ranked by the amount of ACE2 gene expression is given here on the right. I find it interesting that ACE2 gene expression levels in the lung are very, very low compared to other tissues, having some researchers actually speculate alternative receptors for viral binding. Once we start digging into the data, we actually might have evidence that this is true. Upon viral binding to its receptor, it causes a conformational change in the viral protein, which facilitates viral envelope fusion with the cell membrane through the endosomal pathway. The virus then releases its RNA into the host cell. This genome RNA is translated into viral replicase polyproteins PP1A and 1AB, which are then cleaved into small products by viral proteinases. The polymerase produces a series of subgenomic mRNAs by discontinuous transcription and finally translated into relevant viral proteins. These viral proteins and genome RNA are then subsequently assembled into virions in the ER and then the Golgi, which are then transported via vesicles and released out of the cell where they can go and infect other cells with ACE2 receptors. As you can well imagine, the host cell doesn't take this viral invasion lying down. Primary inflammatory responses occur very early after viral infection, primarily initiated by the innate immune system. These responses are mainly driven by active viral replication, viral-mediated ACE2 downregulation and shedding, and also a host of antiviral responses, which can lead to increased cytokine and chemokine production and cellular damage through apoptosis, which is also known as programmed cell death, and or pyroptosis, which is a cell death that results in elevated inflammation in the surrounding tissues. SARS-CoV-2 infection also causes increased secretion of the inflammatory molecules interleukin-1b, interferon gamma, IP10, MCP1, interleukin-4, and interleukin-10. Most patients can tolerate primary inflammatory responses at the stage of infection with a positive outcome of viral load reduction or even viral clearance, followed by a receding of inflammation. Secondary inflammation responses begin with the generation of adaptive immunity and the appearance of neutralizing antibodies that further diminish viral replication. However, the appearance of these antibodies can trigger immune cells that recognize this viral-bound antibody complex and can cause severe lung injury from aggressive inflammation. It is this uncontrolled pulmonary inflammation that is likely the cause of fatality in SARS-CoV-2 infection. Consistent with a possible inflammatory storm associated with disease severity, ICU patients with severe disease had higher plasma levels of inflammatory markers interleukin-2, interleukin-7, interleukin-10, GCSF, IP10, MCP1, MIP1A, and TNF-alpha. It is hoped that by investigating the way the host cells respond to this virus, we can identify or at least develop therapeutics that can either boost the body's defenses against the virus or inhibit the aggressive immune system that is responsible for so much tissue damage that can lead to the death of the infected individual. A list of antiviral agents proposed for use against the SARS virus shortly after the 2002-2003 pandemic is given in this table. You'll notice that most of the proposed compounds act directly against the infecting virus, although this can be tough given the virus's ability to mutate and evade pharmacological intervention. There have been efforts to boost the body's own immune system, as with giving patients different forms of interferon as shown here. That could help clear the virus or inhibit further infection, but this has been met with limited effectiveness. As far as I know, none of the compounds listed in this table are being widely used to treat the current epidemic, necessitating the development of new therapeutics and ways to attack the infection. Again, the more we know about the virus and how it causes its host to respond to the infection, the more likely we'll be able to boost cellular defense mechanisms that are inhibited by the virus and quell aggressive immune responses that lead to tissue damage. Unfortunately, there is little to no host response data in the public domain on the current strain of SARS-CoV-2 because it's so new. So we looked for a related strain we could use as a surrogate to try to understand the host response to infection. There are 7 known types of coronavirus that affect humans. All types cause upper respiratory symptoms like sneezing and coughing and may even cause fever. Most types of coronavirus are relatively harmless, like the common cold. The 3 strains given at the top, SARS-CoV, MERS-CoV, and the current strain, SARS-CoV-2, produce more severe symptoms than the other types of coronavirus and have been responsible for viral pandemics. All of these viruses are similar at the genetic level, but the CoV and the CoV-2 strains show the most homology at 79%. These 2 strains also have a common animal host in bats, target the same ACE2 receptor on the cell for viral entry, and produce similar symptoms in the infected individual. It should also be noted that the SARS-CoV strain is more lethal than the SARS-CoV-2 one, but less severe than the MERS form. It is because of these similarities that we decided to use studies looking at the host response to SARS-CoV in our meta-analysis to try to understand the current epidemic. In this section, we will go over collecting the data used in this meta-analysis and preview some of the tools used to gather this information. I am starting out by showing you the screenshot of the website Gene Expression Omnibus, or GEO for short. This data repository is where most of the data that is generated by federally funded grants is located. Right now, there's approximately 130,000 data sets deposited in this repository, consisting of approximately 3.5 million samples. As you can imagine, there's a lot of potential gold in these billions of dollars worth of research information that is available to anybody with internet access. The problem is finding those studies that are relevant to your analysis and then devising a way to easily combine all this information to give you a single picture. The GEO site does allow you to search their database by a keyword search. For this example, I've used the term coronavirus infection since we are investigating the host response to this current epidemic and are seeking studies that reflect this type of infection either directly or through a related virus. After hitting the search button, it shows 1,551 geo-entries that have the term coronavirus infection somewhere in the description. We can then click on this, and now we can look for studies that fit our purpose. In this case, we are searching for gene expression studies that use the related SARS-CoV strain mentioned in the previous background video as a surrogate to try to understand the body's response to the current epidemic. We can click on those entries, find one down here, SARS-CoV, click on that. Now, this is giving us more information on this particular study, and we also now have access to download all the samples that they used in this particular entry. Downloading all the studies you plan to use can take a considerable amount of time and memory, and you would still need to analyze them yourself and devise a way to combine all of these data, given that many of them were done with different technologies with different sensitivity. This is not an easy process, but it can be done. I decided to take an easier route by choosing a data repository where they already have downloaded and analyzed these data for you. There are plenty of sites that attempt to collect all the abundant research data available in the public or private domain besides the one I used in this analysis. Some of these data sets have free access, like the excellent R2 Genomics Analysis and Visualization platform that has collected a ton of cancer-related data among other useful phenotypes. I chose to use the commercial base-based correlation platform because not only can I easily identify and download results from my studies of interest, but I can also use the same program to find drugs or compounds that can affect the results of my analysis, which can also impact the disease itself. We'll have more on this later in future sections in this series. Also, the parent company for this platform, Illumina, jumped at the chance to be part of this project when I mentioned to them. And I thank them for all the technical support they've given me throughout this project. Thank you, Jim and Joe. Just like with Geo, we can search this database using a key term. Again, I'm going to use the same one we used on our Geo data set, our Geo database, which is coronavirus infection. And you'll notice as I type, phenotypes start popping up. Again, this is an annotated database. So Illumina has already put these key terms associated with all their entries. So I can select this phenotype. And now I have 37 studies to look through. I can look through all these, or I can even filter it even farther. Say I only want to look at those studies involved in measuring gene expression, and I only want to look for samples that have been derived from primary tissue. We chose to focus on the tissue rather than the cell culture because we wanted to not only see how the infected cells respond to the virus, but also the resulting immune system, since it's responsible for so much tissue damage in severe forms of the infection. I do think there's plenty to learn at looking at cells, and I could absolutely look at that aspect as well, but you have to start somewhere. Once we apply these filters, we are now down to 21 studies. We decided early on that we wanted to make this a focused meta-analysis for this first pass. So we looked for studies that measured the host response early in infection. We felt that if we could understand how the body responds initially to this invasion, we could possibly alter this response to prevent the virus from gaining a foothold in the first place. We can click on these entries to see if any of their biosets or comparisons used in these studies match our criteria. In this first study, we can see that this group challenged monkeys to different strains of the SARS-CoV virus. We can then choose to include those biosets that examine the lung response to the virus at day 1 of infection. So these first 3 up here. By clicking on the plus sign to the left of these biosets, I can include this in my meta-analysis, and they will show up in my meta-analysis app after I choose them. I want to be clear right now is that we are not downloading the raw data from these files, but using the change in gene expression compared to the control given in the bioset. The software considers any gene with a p-value less than 0.05 and an absolute fold change greater than 1.2 to be significant. The number of genes associated with a significant change is given by the number on the right of each data set. After going through the below studies, I was able to find 6 entries that contain 13 biosets that we can now view and download using the meta-analysis app. Given on this first tab are the biosets we picked to be included in this meta-analysis. You'll notice that all of these studies are looking at the transcriptional response in the lung to SARS-CoV 1 day after viral infection as compared to mock infection. The first 3 biosets listed were done in monkey models, while the rest of these were done in mice. What makes this data set we've constructed extremely useful is that we compare the changes in gene expression due to infection to the same benchmark in all comparisons, which is normal, healthy lung. This gives us an anchor we can use as a reference to compare all these studies together. We can also normalize these biosets further by putting all of these gene changes on the same scale, which we will discuss in this next tab that looks at the genes that were altered in all the biosets. Given below on this tab are the genes whose expression levels were altered due to SARS-CoV infection, ranked by the number of biosets that gene was observed to change in. Each column on the side of these genes represents the biosets we picked, while the height of that bar represents the change in expression of this gene to our mock controls. The higher the red bar, the greater the increase, while green bars going in the opposite direction would signify a decrease to control. We can see that most of these top genes increase due to infection. Although when taken as a whole, most genes in this analysis on average went down with infection. If we look at some of the names on these genes, we can see almost all of them have been implicated in the inflammatory response. There are lots of interferon-stimulated genes in this list. We can also see quite a few of these OAS genes. They tend to pop up at the top of this list. These genes have been shown to be induced during viral replication and can have positive effects, like binding viral RNAs and trapping them in stress granules, or negative ones, like the same gene inhibiting type 1 interferon responses that could lead to chronic viral infection. We can examine what these genes do in each study by clicking on a particular gene to get more information. This top gene, CMPK2, is one that I never heard of, but doing a search on it reveals that it is involved in synthesizing an antiviral ribonucleotide that can gum up viral RNA and DNA synthesis, which is a relatively new finding for us humans. It also works with a gene commonly called viperin, which is also ranked pretty high on this list, right there. We can go further into this gene to get more information. We can get the probability that it's different than the control. We can get the fold change information given over here on the right. And more importantly, we can get the Illumina score for that gene. You'll notice that all of this fold change activity varies widely, going from 1.6 fold to 51. But when we look at these Illumina scores, they are all in the 90s. The Illumina scoring system is based on the magnitude of change for that gene compared to the other genes in that data set. The greatest change in gene expression is ranked as 100, and everything else is scaled down to 0 from this number. It is this scoring system that allows us to combine all of this information together into one data set. We can then export all of this information with this button here and use the Illumina score and fold change information to make our own database. The correlation engine platform will only allow you to download the top 5,000 genes based on the cumulative Illumina score across all data sets. what we get is a spreadsheet that looks like this. All of the biosets that we used in our study are given at the top of this table. Down below, we can see the individual genes, its overall gene score, which is based on the cumulative, the sum of all the Illumina scores across all the data sets, its individual Illumina score for that particular comparison, its p-value, and its fold change. We can then extract the Illumina scores for each data set and use a program developed by a former student of mine, Mehdi Ahmadi, that will make this score positive or negative based on the direction of the fold change. What we now have is a data set where all the change in gene expression is on the same scale, and we can tell the direction and magnitude of this change with this score. In the next video, I'll explain all the files we have available for download, and then we will start getting into analyzing and interpreting this data set. If you would like to get started now, all of these files are open to download at my GitHub account at the following URL, or follow along with the series to see how I would analyze these data. Thanks for viewing.